Welcome to this lecture. In this video, we'll discuss glycogen metabolism. Now, glycogen metabolism has two parts. We have glycogen synthesis and glycogen breakdown. Now, by the end of this video, we will discuss both of them, both the synthesis part and both the breakdown aspect of it. But before we go into that, we need to ask ourselves this question: what is glycogen? What is what? Glycogen. Please don't mind the noise. What is glycogen? Now, glycogen is simply put, it is the storage form of carbohydrate in animals. The storage form of carbohydrate in what? In animals. Storage form of carb in animals. Now, what about the storage form of carb in plants? The storage form of carbs in plant, that is carbohydrate in plant, is what? Is starch. Then, what then is cellulose? Cellulose is just a structural component of cell wall of plant. Okay, so both glycogen, starch, and cellulose, they are all polysaccharide. They are all what? Polysaccharide. Okay, just that glycogen now is storage form of carbohydrate in plant. In animals, starch is storage form of carbohydrate in plant, while cellulose is a polysaccharide that is just a structural component of the cell wall of plant. You get? So that is one thing you should take note of. So all of them are all polysaccharide. Now, before we go into the synthesis again, I would love to ask you another question. If we say glycogen is storage form of carbohydrate in animals, which part is it stored in? Like, which part of the animal, which part of humans store glycogen, or where is glycogen stored? That is what I'm trying to ask you. Where is glycogen stored? Where do we store glycogen? Where do we store glycogen? So I believe you will be thinking something about the liver. If you are thinking about the liver, then you are correct, because liver is one of them. Number 2 is the muscles. Okay, number 2 is what? Muscles, skeletal muscles. Okay, so these are the two major places where glycogen is what? Is stored. Now, between these two, between the glycogen in the liver and the one in the muscle, which of them do you think can actually maintain falling blood glucose level? Let's say, for example, you have a patient who is fasting. Which of these glycogen stored in the liver and the muscle, which of them do you think can maintain falling blood glucose level? Because when the patient is fasting, the plasma glucose level begins to fall. So between the liver and the muscle, which of them do you think can maintain falling blood glucose level? Now, if you are thinking the liver, then you are correct, because the liver has the ability to give back its stored glycogen into the blood. But the muscle, I would say, is selfish. Now, there's a reason why, as we proceed in this lecture, you get to understand that there is a particular enzyme that is present in the liver, but it's not present in the muscle. It is because of that enzyme, that's why the liver can maintain falling blood glucose level, while the muscle cannot maintain falling blood glucose level. As we proceed, I will tell you the name of that enzyme. Okay, so you just put that at the back of your mind. Now, having known this, in order for we to understand glycogen synthesis, we need to put at the back of our mind that glycogen is highly branched. It means that we will need to synthesize straight chain, and branch chain. We need to synthesize both straight and branch chain. So we need to synthesize this one I'm synthesizing now is straight chain. Then when I add to this place, this one is this is a branch point. Okay, this is a branch point, this is a branch point, and also, all right, now this is straight bond, a straight bond, a straight bond, a straight bond, a straight bond. Okay, this also is a straight bond, it's a straight bond, but this particular point is the branch point, or the branch, let me just say the branch point or the branch bond. You get it now? So what I'm trying to stress here is that just because this entire stuff is more like a branch does not mean that all the bond are having branch bond, because branch bond is different from straight bond. Straight bond is referred to as alpha 1 4 glycosidic bond, but your branch point bond is alpha 1 cis glycosidic bond. So what am I trying to tell you here? I'm trying to tell you that this one here, the bond here is alpha 1 4, 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 the bond here is alpha 1 4. This is also alpha 1 4, but this particular bond at this branch point here is what? Alpha 1 cis glycosidic bond. It's important for you to know this before we start. Again, this is what I'm trying to say. I'm trying to say that, let me use circle this time around. Imagine that this is a circle, and each of these circles represent glucose. Each of these circles represent what? Glucose. Each of these circles represent glucose. Then here is our branch point. Here's our branch point. We can have multiple branch points. Okay, we can have multiple branch points. This bond is alpha 1 4 1 4 1 4 1 4 1 4 1 4 1 4 1 4 1 4. You get this one is 1 4 1 4 1 4 to whatever anywhere you are going to. But you see this point where this branch from, that is called what? Alpha 1 cis. This is called what? Alpha 1 cis bond. So take note of that. It's very, very, very important for you to know that, because as we synthesize the glycogen, we will need to synthesize straight chain, and we also need to synthesize branch points. Okay, so having known that, the next question I want to ask you is, which enzyme do you think is required for the synthesis of glycogen? Because now we've entered glycogen synthesis now. Which enzyme do you think is required for the synthesis of glycogen? Now, I will make it easy for you that there are two main enzymes. One to synthesize the straight chain, and the other one to synthesize the branch point. The one that synthesizes the straight chain is called glycogen synthase. It's called what? Glycogen synthase. The one that synthesizes the branch point is called what? Branching enzyme. It's called what? Branching enzyme. Okay, so it's very, very easy. Glycogen synthase synthesizes the straight chain, meaning I can say that glycogen synthase synthesizes what? Alpha 1 4 bond, right? Alpha 1 4 bond. While branching enzyme is responsible for what? Alpha 1 cis. Do you get it now? So glycogen synthase for the straight chain and branching enzyme for the branch point. Does it make sense? As we proceed now, we'll get to know how they actually do it. Okay, now since we know that the starting molecule for glycogen synthesis is glucose, because glucose is the monomer, right? It means that if we want to synthesize glycogen, we must get our glucose. Now, I will picture that this is our liver here. I will picture the liver here. Okay, this is our liver. Then glucose is going to enter this liver like this. So this is glucose. Glucose has entered the liver. And I believe you know that the first thing to do whenever glucose enters a cell is to trap the glucose in that cell. So that's why the next thing we will do is to form what we call glucose 6-phosphate. But the idea is that if we continue in this glucose 6-phosphate pathway, we will end up doing glycolysis. But do we want to do glycolysis? No, we want to do glycogen synthesis. It means that we are going to move the phosphate from carbon 6 to carbon 1. We now got glucose 1-phosphate. Okay, remember this enzyme here is called exokinase. This enzyme here will be a mutase enzyme. It will be a what? A mutase enzyme. So you can just call it glucose 6-phosphate mutase, because it moves the phosphate from position 6 to position 1. Okay, now that we've gotten glucose 1-phosphate, we can now synthesize glycogen. Please bear in mind that glycogen is synthesized in the cytosol. Anytime you are synthesizing something, we usually synthesize it in the cytosol. But anytime we are breaking down a molecule, we break it down in the mitochondria. Does that make sense? Anytime you hear they are doing oxidation, usually in the mitochondria. Anytime you hear they are doing synthesis, usually in the what? In the cytosol. But an exception to that, where we are breaking down something, is glycolytic pathway, because glycolytic pathway occurs in the cytosol. That's just by the wayside. All right. Now that we've gotten glucose 1-phosphate, before glycogen synthase will start working before glycogen synthase will start working for you, there are two conditions. One, you must have a primer. A primer must be available. And number two condition, so these are the two conditions given to us by glycogen synthase. Okay, I'm just doing this so that you can get the concept. Number one, a primer must be available. Number two, glycogen must be activated. The glucose 1-phosphate must be activated. So let's try and fulfill this one first. Let's try and activate the glucose 1-phosphate. So we have glucose 1-phosphate. In order to activate it, we need to add what we call we need to react it with UTP, uridine triphosphate. When we react it with UTP, we will get what we call UDP glucose. UDP what? Glucose. UDP glucose. UDP glucose with pyrophosphate as a byproduct. Okay, pyrophosphate as a byproduct. So now that you've gotten UDP glucose, there's a particular enzyme that does this reaction. There's a particular enzyme here. The enzyme is called UDP glucose pyrophosphorylase. UDP glucose pyro. Let me just write it here. The enzyme is called UDP glucose pyrophosphorylase. Okay, this is the name of the enzyme that does this particular reaction. Okay, take note of this name of this enzyme. Now that we've gotten UDP glucose, the next thing to do is to start picking the glucose from this molecule and use it to synthesize the glycogen. Okay, that is the next thing for you to do. You need to start picking the glucose from here and use it to synthesize your glycogen. Again, I said that there are two main enzymes in glycogen synthesis: glycogen synthase and branching enzyme. Okay, now I said there are two conditions that glycogen synthase will give to you. One, you must have a primer, and number two, the glucose must be activated. So I said that glucose enters the cell and get converted to glucose 6-phosphate. Then it's now converted to glucose 1-phosphate by a mutase because we want to synthesize, we don't want to break down. Okay, now that you've gotten glucose 1-phosphate, you need to activate it using UTP, and we got what? UDP glucose. Now that we've gotten UDP glucose, the next thing is to ensure that our primer is available. Now, what is a primer? A primer is a short strand of, it's a short sequence of DNA, or a short molecule of DNA, of glucose rather, not DNA, of glucose, together with a primer protein. So imagine this structure now. Imagine we're having this. Let me just draw this. Imagine that this is just 4 residue of glucose. 4 residue of what? Glucose. Right? It's more like the previous glycogen that was broken down, but it was not broken down to the end. Okay? This is 4 residue of glucose. Okay? Attached with what we call, let me use another color. Attached with a primer protein. Imagine that this is a primer protein. Okay? So this is 4 residue of glucose, right, attached with what? The primer's protein. That's what we call the primer. So a primer consists of a short glucose residue, right, together with what? Primer's protein. Now this primer's protein is what we call glycogen. So take note of it. When they ask you what is glycogen is the primer's protein, but the primer itself consists of the short residue of glucose plus the primer's protein. So now again, I want to ask you, what are the 2 requirements given to you by glycogen synthase enzyme before it can start synthesizing glycogen for you? Number 1, activated glucose must be available. And number 2, you must have a primer. Now we've gotten our primer. Here's our primer. Our primer consists of glycogen and a short residue of what? Glucose. Okay, now let me write out our primer again. Here is our glycogen, and here is our what? Okay, let me do it the other way around. Here is our, I want to do it like this. Here is our primers protein, right? That is glycogen. Then here is our what? Short strand of glucose. Now, you know what will happen? Imagine here is, just picture this diagram, that here is UDP glucose. Then inside UDP, here is UDP rather. This thing I'm drawing now is UDP. Then here is our glucose. Okay, or let me use white. Or let me just use the yellow. Here's our glucose. Now you know what will happen? Glycogen synthase, which is the first enzyme, we start picking glucose from this molecule and add it to this. We synthesize glucose like this, keep on adding it. Okay, when the glucose has gotten to a particular length, let's say about 10 or 15 lengths, it will now stop. Then the other enzyme will now come in and create a branch point. So the function of glycogen synthase is to make alpha 1,4 bond. Is to make what? Alpha 1,4 bond. When you get to about 15 glucose residue, it will stop. Then the second enzyme, which is branching enzyme, will now come into play. Second enzyme is what? Branching enzyme. Branching enzyme, look at what branching enzyme will do. Branching enzyme will make a cut here, like this. We make a cut here. I'm just taking that one as a hypothetical case, right? We make a cut here and transfer about 8 residue of glucose. Look at what it will do now. So we are going to transfer that section to somewhere here like this. So this is how we do it. Okay. So now I'm not permitted to clean from here. I'm getting it now. So that's how it does it. So you can see it. So now we've gotten our branch point. That's what the branching enzyme will do. So in essence, glycogen synthase makes straight chain, while the branching enzyme make what? Branch point. Now that we've gotten this branch now, I can say that here is what? Alpha 1 6, right? Alpha 1 6, okay? But this one is what? Alpha 1 4 1 4 1 4. Is that also? Now, after the branching enzyme has done its work, then glycogen synthase will now start elongating. Will now say, oh, you break out the one I did before, I will make more point again. We'll now be making more glycogen. It will also make at this point here. It will make at this point. Okay, you know what will happen is branching enzyme will now come in, make another cut. Let's say make another cut somewhere here like this. You know what will now happen? We are going to attach it somewhere else. So we'll do something like this. We'll now attach it like, let's say we're attaching it here. So it will now be like this. Okay, then I can erase this part. Here you are seeing it now. You can see that we've made what? Another branch point. Okay, we've made another branch point again. So you can see that's how the molecule will keep on going like that. Keep on going. It can choose to synthesize. It can choose to also cut here. It can choose to also cut here, like the branching enzyme now, and create another branch somewhere here like this. You are getting it. So in essence, glycogen synthase make straight chain, while branching enzyme make what? Branch point. Okay. So in this video, I've been able to explain to you that there are two main enzymes in the synthesis of glycogen. There are two main enzymes. Number one is what? Glycogen synthase. Glycogen what? Synthase. And number two is what? Branching enzyme. Branching enzyme. And I have been able to tell you that glycogen synthase has what? Two requirements. Number one, the glucose must be what? Activated. Glucose must be activated. Number two, glucose must be activated. And number two, a primer must be available. All right, so we'll end it here. In the next video, we'll continue from glycogen breakdown. Then we'll move to glycogen storage diseases.